Hello, and thank you for tuning in to Answers from the Lab, where we share Mayo Clinic knowledge and advancements on the state of testing and science from laboratory leaders and the people who are making it happen behind the scenes. I'm Dr. Bobbi Pritt, Interim Chair of the Department of Laboratory Medicine and Pathology at Mayo Clinic in Rochester, Minnesota. With me today is Dr. Bill Morice, the President and CEO of Mayo Clinic Laboratories. This is our weekly discussion with Dr. Morice, in which we learn about updates in the field of laboratory medicine and pathology. With us today is Dr. Elitza Theel, the Director of the Infectious Diseases Serology and Sexually Transmitted Infectious Laboratories. Dr. Theel has been a regular on our podcast, and it's great to have you back. Welcome back, Ellie.
Thank you so much. Happy to be here.
So we were just at a meeting together, the Mayo Clinic Infectious Disease Conference, and we learned about clinical practice updates, new developments in the field. This is a wonderful meeting we do every year with our partners from infectious diseases. There's always so much to talk about. So I'm really glad that you joined me today to talk about some of the things that you had presented about how things that relate specifically to laboratory medicine and some of the inspiring work you're doing in your laboratory.
Yeah, no, it's a really wonderful conference, really high educational value for both new clinicians and, practicing ones that have been around for a while. So it was great to be there.
And it's in Hawaii. How could you go wrong?
And it's in Hawaii. Yes.
So you specifically presented on the topic of at-home and in-clinic specimen collection for sexually transmitted infections, STIs. And that's such an important topic. We know that patients want to be able to collect their own specimens, either in the comfort of their own home or in a private clinic setting. So can you explain how this type of testing works and what makes it innovative compared to our traditional collection methods?
Yeah, so I think this is an area that's going to expand quite a bit in the next few years. But there's, a number of different types of STI tests right now. There's the classic test where the clinician collects a sample and gets sent to a central clinical laboratory for testing. That's the one we're all familiar with. But increasingly, companies are getting FDA clearance for self-collection of samples in a clinical setting. So patient comes in, see their clinician gives them a swab, they swab themselves, and then that sample gets sent to the central clinical lab. So that's the second type of test. Mm-hmm. The other type of test are self-collections and testing at the point of care. So a patient comes into an STI clinic, for example, they collect their throat or rectal or vaginal sample, give it to the clinician tests it at the point of care, and they get a result within 30 minutes, and they can rapidly implement treatment if needed. More excitingly, there is now the opportunity for clinicians to order a self-collection kit to be sent to the patient at home. The patient collects that sample, sends it back to the lab, the central lab, for testing. So that's another option. And then more recently, there are now 2 FDA cleared over-the-counter testing kits where the patient can purchase the test, collect it at home, and then test the sample themselves at home. So there's one for syphilis that was recently released, and then an HIV antibody test that can be done at home. So really exciting times, but it's really important to know that these are all considered different types of tests in the eyes of the FDA. So validation, verification, and implementation, there's just different things to consider with all of those.
Sure, many different implications, especially for us in the laboratory, if we're considering offering a test for patients at home, lots of things for us to consider. But I would imagine that this will improve the availability of STI testing. What are your thoughts on that?
I think it's wonderful from the patient perspective. So when you think about STIs, like it's a private, sensitive topic, an issue. So the patient being able to collect the samples themselves, collect them at home. So not having to go into a clinic, it provides privacy, autonomy, really, in a sense, potentially increased availability of testing, especially if they can purchase it over the counter. So the idea being that if, more testing is available, we can more rapidly identify these infections and treat them. But there are some limitations to that, primarily access to care. So we can do the testing, but we still need that access to care to treat those positive patients.
Yeah, so I would assume that that's some of the challenges, or one of the challenges amongst others. So you've touched on the benefits, but can you tell us a little bit about these challenges associated with these different types of specimen collection and testing methods?
Yeah, so the home testing, I think, is the biggest issue. It's, again, really good for patients, but a number of key challenges. We talked about, you can test, but you still need access to care. So getting that access can be challenging for some patients, as we mentioned. The other thing is that for a number of these STIs, they are tracked, right, by public health entities. So in a clinical setting, if we get or in the laboratory, if we get a positive result, that result, through various different channels, is reported to our public health colleagues, who will then do follow-up communication and reach out to any potentially exposed individuals or partners.
Sure.
The problem with home collection is there's currently no way to automatically report results to public health entities. So we run the risk of having positive patients, but there being no follow-up to potentially exposed partners. So that's one challenge that I think really needs to be worked out before this is as effective as it could be. The other issue that has come up is there are a lot of home collection kits that you can purchase online. I actually did a mini little Amazon search. There's over 50 different STI tests that you can purchase right off of Amazon.
Wow.
Trick is, the vast majority of these are not FDA cleared. We don't know what laboratory is performing the testing. We don't know what tests are being used to perform those tests. And so the accuracy of these home collection kits and the results is a little bit questionable, or I guess unknown, we should say unknown. Sure. So there's some deceptive marketing out there, a lot of home collection kits, but those samples still go to a lab for testing, and we don't know how well that testing performs. So that's a bit of a challenge and something that we need to consider as a laboratory, in the laboratory community. And then for clinicians, if a patient comes to you and says, I did a home test, probably good practice to confirm any potential positive results before starting treatment.
Sure, that makes sense. Yeah, I'm just thinking of the importance of public health and tracking infections, infection rates. How else are you going to be able to identify trends? For example, we know congenital syphilis rates are increasing. That's all because of this reporting mechanism. And that would, if everyone's doing testing at home, they may not want to report infections. So lots of implications there.
Yeah, I think it's one of the biggest concerns with home testing specifically.
Yeah. Well, are there any other recent advancements in STI testing that you find particularly exciting or promising for the future of infectious disease control?
Yeah, like I said, I think it's wonderful that we're getting more and more FDA cleared kits. So again, we have one for syphilis and one for HIV for home testing. The challenge with STIs, though, is that we detect, like, gonorrhea, chlamydia, mycoplasma genitalium. We detect those using molecular-based assays. And right now, there are no affordable, accurate, single-use molecular assays that are really cost-effective for home testing. So I think one of the exciting things in the field is seeing where that progresses. Will we get to a point where we can have an at-home molecular test? I know there's a number of these sorts of assays under development, so I'm kind of excited to see where those go, and hopefully in the next, 3 to 5 years, we'll have the opportunity to use these sorts of tests at home for these other more common infectious STI tests. or pathogens.
Yeah, that is really exciting. And I really do think it'll improve access for all the reasons you mentioned. And also just the fact that some people don't live close to a clinic, and getting to the nearest place to do a collection could be an hour, 2 hours drive. So potential benefits, but with those concerns, but very
Yep.
exciting. Well,
Yeah.
are there any gaps that you'd like to talk about then in STI testing? We kind of touched on a few indirectly, but what are you currently seeing?
I think one of the biggest gaps or problems is associated with antimicrobial resistance for some of these pathogens. So we're really seeing an increase in resistance rates for a number of the STIs, really gonorrhea and Mycoplasma genitalium worldwide, not necessarily just in the US. So for gonorrhea, currently the first line drug of choice is ceftriaxone, and thankfully we have very low resistance rates to ceftriaxone, less than 1%, which is great. But that's not the case worldwide. Australia, for example, has a pretty high rate of resistance to ceftriaxone. And one of the things we're lacking are diagnostic tests to detect these resistance markers. So a number of companies have started working on developing genotypic assays to detect some of these resistance markers. So for example, what they're doing in Australia right now for gonorrhea in particular is they are using an assay or various different assays to look for fluoroquinolone resistance. And if they see resistance, they will go ahead and treat with ceftriaxone. However, if the fluoroquinolone markers for resistance are negative, so in theory susceptible, instead they'll treat with a fluoroquinolone in an effort to decrease the risk of developing ceftriaxone resistance. We don't really have those assays available in the United States as of yet. Again, low rates of ceftriaxone resistance at this point. The other organism to just talk about with respect to resistance is Mycoplasma genitalium. Increasing rates of macrolide resistance at this point. So a number of companies have developed genotypic assays to look for those resistance mutations in the 23S ribosomal RNA for Mycoplasma genitalium. The idea there being that patients right now are treated with doxycycline to start to decrease that bacterial load. And the idea would be that if you have a macrolide resistance assay available, you would test the sample. If it is resistant to macrolides, you would add on a quinolone to your doxycycline treatment, whereas if the macrolide is susceptible, you would treat with azithromycin, for example. So the availability of these sorts of genotypic susceptibility assays, I think, is going to be more and more important in the future so that they can better guide antibacterial therapy. But again, at this point, not a lot of options available on the market, so it is a gap.
Yes. Well, hopefully they will raise awareness of the problem, and then we will hopefully see more companies developing assays that they put through the FDA, and then we'll have tests we could use in the US that are FDA approved or cleared.
Yes. Yeah. One of the challenges is the way that we test for STIs. We don't isolate organisms anymore. It's all molecular. So we really need genotypic based assays to look for these markers because we can't easily use phenotypic methods to look for this.
Yeah, that makes sense. I remember learning about Neisseria gonorrhoeae and how it's such a fastidious organism. It doesn't grow well. And right, who even does culture for that? Really, it's not considered the gold standard. The nucleic acid amplification testing is the gold standard. So I suppose detecting resistance mutations means that there are readily available or readily identified mutations that are clearly associated with resistance.
Right. Yep. For a number of the antibacterials, that is the case.
Yeah. Well, this has been very interesting, and we'll look forward to hearing more. And I understand that you're going to be bringing live some tests in the future for at-home or in-clinic collection. So we'll have to have you come back and tell us about how that goes at a future podcast.
Happy to come back and talk about our progress.
Wonderful. Well, thanks again, Dr. Theel, for joining us.
Thank you so much.
Thank you for tuning in to Answers from the Lab. Be sure to subscribe to this podcast and tune in every other Thursday for new episodes.